And me and my teammate had swung around and just cracked into each other, and I was sitting there like, where am I? Yeah. Oh, I'm on the field, right? It's bizarre, just complete disorientation. Yeah, like, I'm not taking out. Moving on. You want to go to bed? Some people like through that though, and I'm like, that's not the way that. Full out now. No, brain damage. Yeah. To some people it is. Some people no.
All right, I think we have everybody that's going to take the test that has taken the test. So I always like to share with you like the distribution of scores, just so you kind of, I don't know, see what everybody else is doing. You're not the only one that got whatever score you got. This was overall, this was a pretty good start. The mean was a 76 when it was all said and done. So that's a pretty decent start. Nobody got them all right. Had a few people that only missed one, a few people that only missed two. Obviously, some of you, if you did great, just keep doing what you're doing. If you didn't do as well as what you were hoping, then now is the time to kind of think about, well, what things could I change before the next exam? So do I need to study with somebody? Do I need to get a tutor? Do I need to come to office hours more often and just make sure I'm understanding things? Do I need to pay more attention to learning objectives? Do I need to get more sleep? Whatever it is, don't just assume that if you don't change anything, you're just going to do better next time without changing anything. So I would encourage you to come and take a look at your exam. I've had a few people trickle in and do that. I'll be in my office after class today, if that works for you. Let's go over, though, a few of the most commonly missed questions. So there are four or five here that I thought that we would go over. So certain bacteria release endotoxin when the cell dies. Remember, endotoxin, that's part of that lipopolysaccharide. That's the outer membrane, which is considered part of the cell wall of gram negatives. So I think there was some confusion over whether cell membrane or cell wall. There's a plasma membrane, and then the cell wall, LPS, lipid A, part of that cell wall. Oops. And I think that one was the most commonly one missed, and it was somewhere between, like, I forget it. It depended on which form you had, but somewhere between 25 and 30% of you got that right. So there wasn't any one question. There was one question that everybody got right except for me. So when I looked at my Scantron, I was like, I think I made a mistake here. So I had to go back and fix some things that your score reflects what I fixed already. But so there wasn't any like one question that was super hard that like only a few of you got right. I knew this one would be tough. If you were, which of the following would be considered the most desirable resolution? So the best resolution is the shortest distance. And if we talked about micrometers, that's this micro m, nanometers is the smaller unit than the micrometer. So correct answer here is B, 2 nanometers. Even 0.02 micrometers, 2 nanometers is equal to 0.002 micrometers. Yeah, so I knew that would be a tough one. All right, some of you got that right. And I knew this one would be hard just because it was kind of a detail of an experiment that we looked at, but it wasn't sort of like the main maybe focus of what we were talking about. So we gotta an exam, we gotta have a few tough ones thrown in. Which of the following is not an observation Pasteur made concerning the fermentation of grape juice? B here, pasteurization kills yeast to prevent spoilage of grape juice. It's the bacteria that spoiled the grape juice, that spoiled the wine. So the yeast is what they wanted to keep there. That's what was doing the desirable fermentation. One more, yes. So the composition of cilia is like that of flagella, and cilia are only found in eukaryotes. Okay, so when I say composition, I'm talking about the structure. What's the protein that it's made of? Okay, it's made of tubulin, where prokaryotic flagella are made of the protein flagellin. Okay, and fimbriae is made of a different protein, pelin. Okay, like the pilus. So, all right. So again, if you have questions, you want to look at your exam, you want to check over what you missed, I'd always certainly encourage you to do that sometime during office hours, or we'll find another time. All right. So when we left off after the exam, at chapter 5, unicellular eukaryotes is kind of a smallish chapter. We did a little bit before the exam, and then we almost finished it after the exam. We talked about protozoa, some characteristics of protozoa, and we talked about a few structures like trophozoite, that's probably new, cyst stage, and pointed those out as part of the malarial life cycle. Talked a little bit about the disease malaria. And then last time we talked through some fungal structures. Hopefully it didn't overwhelm you with that. We're not really going to go through any kind of asexual reproduction or anything, but I did want to talk about at least one fungal pathogen. I'll talk about more at the end of the semester if we have time, but that Coccidioides species is a fungi that when you inhale the spores, then you get this fungal pneumonia that typically isn't too severe, but for people that maybe are immunocompromised or have, you know, some other kind of risk factor, they're just older, our immunity wanes when we're older, then it can be a little bit more serious. How serious is it? When does it occur? As of 2019, there was about 20,000 cases in the US. And where does this occur? This is a Western Hemisphere thing. Doesn't seem to be any cases here in Asia or Europe. Mostly hot, dry places is where we find these spores in the soil. So in the US, most of the cases are in California and Arizona. This Coccidioides, coccidiomycosis is also known as valley fever, that originated in that San Joaquin Valley of California. So most of those cases are there. There's only about 200 deaths of this per year in the US. So it does occasionally occur outside of those zones. There's a couple cases in Eastern Washington. And if you look into this a little bit more in the CDC, there's kind of sporadic cases that pop up here and there. But one important thing to remember about this disease and about fungal diseases in general is that they're not contagious. They're not passed from person to person. So that's kind of interesting. You would think, and that question popped up with me as I looked at the life cycle of these. Remember, it's two species of the genus Coccidioides, and one species occurs kind of this yellow region, and the other species in the orange causes the same disease. So we're not going to worry about the species. But when I looked at this life cycle, I thought, well, it seems like you could get from person to person. So let's look kind of briefly at this. I want to point out a few things. The first thing, you see there's two different sides of this life cycle. This is one of those fungi that can be dimorphic. It can take on totally different shapes or morphs of its cell type. And so in the environment, it depends whether it's in the environment or whether in this case it's in a human. So the spore starts out here at the top, a hyphae, if it's considering, say this is in the soil somewhere, a hyphae grows out from the spore. And when we have a masses of hyphae, remember we call that mycelia or mycelium, a mass of hyphae, filamentous growth. And when those mature, they start to form spores within them. And that particular spore structure is called arthroconidia. Again, these little kind of bumps in the hyphae, those are the spores that develop within there. That's an asexual spore structure. So some fungi do sexual reproduction, and in that case, like two compatible hyphae tips would have to fuse, and that's how they mate. But in this case, we get the reproductive spore just forming, within one filament there, so it's asexual. And then if those spores in the soil are disturbed, it could be, a dust storm or some kind of construction or something like that takes place, and those are aerosolized. And then if a person inhales those spores, then the fungus goes into kind of a different phase of its life cycle. It can actually form these spherules, which are kind of many uninucleated cells within kind of one sphere. And this happens typically inside the lungs. So in the corner there, here's one of those spherules inside some lung tissue. And eventually those spherules kind of mature and release those spores. Now, to me, it thinks, and then if those spores would happen to land back in the earth, okay, then it can go back to the other life cycle again. But apparently they don't release, when somebody say coughs or whatever, they don't release in enough amounts to be able to actually be contagious to someone else. So yeah, just a few terms I wanted to point out there: the hyphae, the mycelia, and just the fact that it's one of those dimorphic fungi. Not all fungi are dimorphic, but this happens to be one of them. So, all right, that's pretty much all I had. Any questions about this infection or any of the fungi in particular? All right, so again, that's chapter 5. There's a little bit of homework with chapter 5. I don't think it's too bad. Okay, all right. So moving on to chapter 7. So this is kind of the last chapter before we get into some of the more, I'm still like dumping lots of information on you, right? But we get into a little bit more conceptually more difficult things when we get into metabolism and genetics. But here we did a little piece of chapter 7 when we talked about growth mediums and such. We want to go back and hit the rest of that chapter. The first topic that we'll explore is what elements exactly do microbes need to grow? And that kind of ties in with the textbook in chapter 8, where that's the chapter called microbial biochemistry. And I did assign just a couple of questions in 8.1 and 8.2. 8.1 is organic molecules and 8.2 is carbohydrates. There is a section on lipids and proteins as well. This is a good chapter to review, especially if you have not had biochem yet. Hopefully it'd be really quick review if you've had biochem, but read through that. And again, there's just a few questions there. It just kind of relates to macromolecules. What are those? So just some kind of terms that I'll kind of assume that you're familiar with. Then we're going to talk a little bit more specifically about how microbes grow. I've kind of referred to that a little bit already, but we'll look more in depth in that. And we're going to talk how we can do, how we can predict microbial numbers mathematically. This is the one time that we do a little bit of math in here. And that includes some growth curves and talking about how to count cells. If you're in lab, you've done a little bit of that already. And then other topics that we'll touch on here, just thinking about the conditions, the environmental conditions that bacteria need in order to grow. And they're quite varied. So we'll look at, different oxygen requirements, pH requirements, temperature requirements, and how that affects growth, and a few other things as well. So hopefully we start to understand that no matter, what the oxygen concentration or whatever the salt content or whatever, there are microbes that can grow there. So they are pretty much everywhere. Not in fire and not in the sun, but pretty much everywhere. Okay. All right. So when we think about the actual kind of elements that microbes need to grow, obviously cells need carbon, hydrogen, and oxygen in really large amounts. Okay. They're just in pretty much everything. They're in every cell structure. Remember, if cells have a carbon hydrogen bond, if a molecule has a carbon hydrogen bond, then that's considered an organic molecule. Okay, it's not just carbon. Something like carbon dioxide is not considered organic. It has to have a carbon hydrogen bond. So again, this is carbon, hydrogen, and oxygen. They're in pretty much everything. Nitrogen, we find a lot of nitrogen in nucleic acids. Nitrogenous rich bases, you might have heard that term before. Lots of nitrogen in amino acids, okay, that make up proteins. So altogether, about 95% of the cell is typically made up of those 4 elements. So a macronutrient is something, I guess I skipped over that. A macronutrient is something that the cell needs in really large amounts. And sometimes different sources will kind of name different macronutrients. This CHOMPS, CHONPS, CHOMPS, so that's kind of an acronym for, I guess, the things that are most commonly referred to as macronutrients. Phosphates, that's, a cell needs a lot of that for its membranes. A phospholipid membrane, for example. Phosphate is part of the energy currency of the cell, ATP, adenosine triphosphate. And then sulfur is often considered a macronutrient as well. It's going to be in some amino acids, as well as disulfide bonds, which are a component of lots of proteins are going to have that particular kind of bond in it. Let's see, this is too big. Okay. Just as important are the micronutrients, no less important than macronutrients. The cell just needs them in really small quantities. A few of these, like iron, sometimes that's considered a macronutrient. Here they considered it a micronutrient. But these are elements that often are needed so that some enzyme functions correctly. So again, trace elements, they're just needed in such small amounts, enough, like what's in tap water would probably be enough. Okay. And then anything organic that a cell needs, we also often refer to as growth factors. Some of these growth factors, bacteria in particular, can make themselves. Sometimes they have to get it from the medium that they're in, but things like amino acids, the electron carrier, NADH, vitamins, for instance, those would all be considered some kind of growth factors that we may or may not have to provide for some kind of bacteria if we're trying to grow it in the lab. All right. So additionally, related to that, we have terms that describe how organisms acquire their carbon and their energy. And this is kind of a third grade version of an autotroph, that term, what it means to be an autotroph and a heterotroph, but I thought it's good to start with that. So troph, that term, or that root of that means nourishment. Or you could say to eat. Sometimes we say it that way. So some organisms, and again, these describe how organisms acquire carbon. Whoops. Okay, so some organisms can kind of do it themselves. Autotroph means kind of they get carbon themselves. They're not reliant on any other organism. So an autotroph gets their carbon from CO2. Okay, just from the air. So all of our plants, algae, some bacteria are considered autotrophs. They get their carbon from carbon dioxide. If an organism is a heterotroph, hetero means other. Okay, so a heterotroph gets carbon from the food it eats. Okay, and that doesn't necessarily mean that an organism has to move around to eat stuff. Remember, fungi, we described them as saprobes. They absorb organic nutrients from dead and dying organisms. But animals, protozoa, a lot of bacteria are considered heterotrophs, again, from whatever organism, other organism that they consume. So and then I think the tricky part is where we add another prefix to these terms that describes how organisms acquire their energy. Okay, so energy source here. Okay, so that's described by two other prefixes, either photo or chemo. And again, over here is our carbon source, whether it's auto or hetero. All right, so we put both those together. A photoautotroph, for example, is an organism that gets their energy from light. In other words, the sun. Okay, and then they get their carbon from CO2. Okay, so that's very popular. All the plants, the algae, cyanobacteria, photosynthetic bacteria do that. Also very popular is the category that we are in, the chemoheterotrophs. We talked about how fungi were chemoheterotrophs a little bit. That just means they get their carbon from stuff they eat, and then their energy source up here is from the breaking of bonds. Okay, the breaking of chemical bonds. Okay, and that can be from organic source or an inorganic source, actually. Okay, but the chemoheterotrophs, that's us, most animals, fungi, and a lot of bacteria. The other two categories here is just made up of bacteria. And these are mostly bacteria that have some very different kinds of metabolisms. They're not bacteria that would live in our body. Most of them would live in kind of extreme conditions. Like these chemoautotrophs are going to get their energy from inorganic sources like sulfur, hydrogen sulfide, iron, that sort of thing. And again, extremophiles, archaea mostly. The photoheterotrophs, they get their carbon from the CO2. Whoops, no, not their carbon from CO2. They get their carbon from organic compounds, and sunlight is their energy source. So again, those are kind of bacteria with some different metabolisms that we're not going to really go into too much, but those are kind of rare. All right, so if we think about, you know, what microbes need, let's also think about how they actually grow. And when I was an undergraduate, this is probably the lab that I remember the most. We took a microscope slide and we put a film of some kind of agar medium on it, and then we inoculated our bacteria on that slide, and we put it under the microscope and we watched bacteria grow. It was not thrilling. But after about half an hour, we focused in on one cell. It would get bigger and bigger and bigger, and then a septum would start to form. And then very slowly it became 2 daughter cells. Okay, so that is binary fission, and that is how most bacteria are going to reproduce, by binary fission. Now, other microbes like protozoa or algae or whatever can do all kinds of different things, but bacteria will typically do binary fission. All right. Okay, and so a parent cell gets larger and splits into 2. And it splits into 2 cells that are clones of the parent. Okay, so what I mean by that is they are genetically exactly the same as the parent. Okay, unless there's some mutation that's entered that comes into the picture there. So when we describe bacterial growth, usually we're thinking about, yeah, the cells get a little bit larger and then divide, but we're mostly thinking about they're growing in numbers. And the way bacteria grow is that they will typically grow exponentially. So that means that the number of cells is going to double after every generation. Okay, so when we talk about generation time, that means the time it takes for one cell to become 2. Okay, so here illustrated on the right, here's one cell, the chromosome replicate. I'm going to talk more about how that happens later on. A septum forms, and eventually here we have 2 cells. So in this case, for example, say that takes about 30 minutes. That's probably fairly standard, but there's a lot of variation in that. And we'll talk about that in a second. Here's another generation, another 30 minutes. Those 2 cells have become 4 cells. Another generation, those 4 cells. Let's pretend that this says 8. So they've drawn 9 there for some reason. I don't know why. And then another, again, generation. So 4 generations there. It's become 16 cells. I said that 30-minute generation time is fairly standard, but it depends a lot. The generation time depends on what species of bacteria we're talking about. E. coli, under ideal conditions where you have lots of nutrients and nice warm place, can have a generation time of about 20 minutes. The mycobacterium, which you may have talked about in lab if you had this morning or maybe tomorrow, they typically have a really, really slow generation time because they have to make that mycolic acid that's part of that acid-fast cell wall. Their generation time may be like 24 hours. So with that difference, we streak E. coli on a plate and we have colonies growing up the next day. With Mycobacterium tuberculosis, you streak those on a plate and it takes about 8 weeks till you have colonies growing up. So it depends on the species to some extent. It's also, the generation time is going to depend on availability of nutrients. Okay, in our gut, E. coli probably doesn't have a generation time quite that quickly, even though it's a nice warm environment with lots of nutrients. Also depends on other physical parameters. Something, we'll talk more about this, say like temperature. Okay, a lot of bacteria are not going to reproduce as quickly if they're in a little, like say, room temperature as opposed to body temperature. But again, depends on the species a little bit there. Okay, so variable generation time. If we looked at expressing bacterial growth graphically, because the numbers, again, is assuming ideal conditions, are going to be growing exponentially, doubling at every generation. If we were to express that, the numbers over time, number of cells over time here on the graph, we'll see that it very quickly, the numbers increase very rapidly. So sometimes to fit that on the graph, it's very difficult. It makes more sense to express that on some kind of semi-log paper, where the y-axis here is logarithmic. So for instance, 10 to the 0, that's the same thing as 0. 10 to the 1 is 10. 10 to the 2, that's the same thing as 100. 10 to the 3 is 1,000. Okay, that's a logarithmic scale. And then the time is just kind of regular time. If we plot bacterial numbers growing over time on semi-log paper, then we get a straight line. And then from that graph, we could say, well, I wonder how many cells I'm going to have at 11 hours. We could go up here and we could figure that out pretty easily, where that would be really hard if we were graphing on kind of regular graph paper. Okay, so let's see what else I wanted to say about that. So if we looked at a growth curve, we see, if we were starting a culture in the lab, say a broth culture, and at certain different times we were taking out and testing the numbers, which we'll talk about different ways to count cells shortly. A typical growth curve would usually look something like this, where there's going to be, if this is right here when you inoculate your culture, there's typically going to be a little bit of a lag phase where the cells are adjusting to their medium. They're figuring out, what nutrients they have to work with, what enzymes they need to make to break down those nutrients. And growth is generally slow right to begin with. But once it kind of figures all that out, then it will start its exponential growth, and we call that the log phase. Okay, so that's where numbers are rapidly increasing. That's when the cells are all going to be very uniform shape and size. That's also going to be when cells are typically more easily killed by antibiotics. And we'll talk much more about the use of antibiotics later, but a lot of antibiotics work on cells that are actively growing. Okay, so they're most susceptible, bacteria are most susceptible to antibiotics during that phase. Once nutrients maybe start to run a little bit low or waste products start to build up, that growth will level off, and we call that a stationary phase. That might continue for some time, and then eventually when the number of new cells that are being reproduced is less than the number of cells that are dying, we get into a death phase, or sometimes we call that a decline phase. But it takes a long time till every single cell in that culture is going to be dead, typically. All right, so 4 phases of a typical growth curve. All right, I want to talk a little bit about calculating cell numbers in terms of trying to calculate, if we wanted to know like how long you had to incubate some cells, how long it would be till you had a certain number of cells. So there's a couple different ways we can think through this. I'll talk about doing it with an equation, and I'll talk about thinking through it. If we had a question like this on an exam, it would be one that you wouldn't need a calculator for. And I've given you a couple of practice ones in your homework as well to think through, and we'll do a question here as well. But there's a couple different ways to think through it. Thinking about using a formula, first of all. So the number of cells arising from a single cell is expressed as 2 to the n, when that small n is the number of generations. Okay, so if we start with one cell, and after one generation, okay, we're gonna have 2 cells. So hopefully that makes sense. What about if you start with 1 cell, how many cells will there be after 3 generations? All right, so the number of cells after 3 generations is going to equal to the number of cells that you start with, that like n initial number, times 2, whoops, to the 3rd power. That's what I meant to say. Okay, so that equals 1 times, and 2 to the 3rd power is 2 times 2 times 2. So that's going to equal 1 times 8, or 8 cells. Okay, so just using, again, small numbers, kind of thinking about how to use an equation to figure out how many cells you're going to have in a certain number of generations. The other way you can think through that, which actually I would kind of encourage you to do it this way, if you just kind of think it through, okay, when I'm starting with 1 cell. That's your time 0. Okay, and then after 1 generation, you get double, you get 2. After 2 generations, you get double that, you get 4. After 3 generations, 8 cells. All right, so you can kind of just figure out I'm gonna have double my number of cells, you know, if you're not working with numbers that are too big. Okay, so let me give you a question that you take some scrap paper and see what you get here. See if you can think through it, and then we'll do it together. But say you start with 1 times 10 to the 5th cells. That's 100,000 cells. Okay, and then see if you can figure out, given the generation time, after 2 hours, how many cells you're going to have. All right, so I'll give you a few minutes to try to think through that. And work with other people if you want. That's fine. All right, so I started to write here just so I don't get goofed up when I try to write and talk at the same time. All right, so using the formula, okay, that's final number equals the initial number times 2 to the number of generations or whatever. Okay, so we said that we started with 100,000 cells. How many generations was that? You had to work that out from the problem. If the generation time is 30 minutes, after 2 hours, it's going to be 4 generations. So that's where I got 2 to the 4th. 2 to the 4th is 2 times 2 times 2 times 2, which is 16. And 16 times 100,000 is about 1.6 million. So 1.6 times 10 to the 6 cells after 2 hours. Now, the other way you could think through it, and I kind of recommend the other way, given that I said I'd always give you numbers that, you know, sometimes, you know, like plugging this number into a calculator is not easy. So given that, I'll give you lower numbers. So this time 0 or generation 0, 100 cells. Okay, after 1 generation, which would be 30 minutes, you're going to double it. You'd have 200,000. Okay, after 2 generations, which would be 1 hour, you're going to have 400,000 cells. After 3 generations, 1 hour, 30 minutes, 800,000. And after 4 generations, that's going to be 2 hours. Okay, same number. Okay, now doing it this way on the right, what always goofs people up is that they'll say, oh, remember time 0 is the number that you're given with. Sometimes people want to call that the first generation. Well, that's the 0 generation. Okay, and then you go 1 generation from there, you double it. Okay, so again, there'd be a couple homework questions to try with that, and you know, we can talk through it if you get stuck on anything. Questions? Hopefully I didn't write anything wrong. Okay. Okay, so you probably will see something like that. So just again, get some practice. Let's talk about ways to measure bacterial growth. And again, if you're in the lab, then you got to do this viable plate count already. I want to talk about a couple of ways to measure bacterial growth that require us to incubate cells. Okay, so one of those is viable plate count. Another one is membrane filtration. So let's look at both of those, and I'll come back to the slide in a minute. All right, so this is a schematic of how to do the viable plate count. What this doesn't show is, so here, assume there's some original culture here, okay? And that's what you're trying to figure out, about how many cells are in that original culture. And usually we express that per mil. So it doesn't really matter what the volume is in that original culture, because again, our answer is just going to be per milliliter. And the idea is that we want to put an aliquot of cells onto an agar plate and let those cells grow up, because we assume that one cell in that original culture will eventually grow into, you know, a colony down here on the plate. So the problem with just taking that original culture and putting that straight on a plate is, assuming that culture is cloudy, there's just going to be way too many cells there that are going to grow into way too many colonies, and you can't count that accurately. So typically we have to make some dilutions. If you did this in lab, we made 1 to 10 dilutions. We took out an aliquot of each of the dilutions, and we actually used a glass hockey stick. Remember, we used a spread plate to spread that around, not your loop, and then counted the number of colonies that grew up, whatever, the next day later or whatever. So we're looking for that countable range where it's not too many and too hard to grow, or not too few that it's not considered statistically significant, somewhere between 30 and 300. So I won't go into how to make that calculation, but taking into account the dilution that you put on the plate, and the dilution of the amount that you put on the plate and the dilution of the tube you use, you can extrapolate back to what the original cell density is in that original culture. Another method that requires incubation is if you have a sample that you know is very dilute already. So most of the time, anytime you take cells from nature or you have some growth medium with cells in it in the lab, there's going to be a ton of cells there, and you would have to dilute it out. But what if you have a culture? Maybe it's a water sample that you're trying to ascertain how many cells are in it. And I've tried to do this with cells from the Grand River. It turns out you don't need to concentrate that. I actually had to dilute it quite a bit to get a countable range. But if you use something like tap water, it's not sterile, but there's not very many cells in there. You're probably going to have to concentrate it. So you would put your sample here that you're trying to figure out. You would know how much volume you're adding here. And you have a membrane filter right here where even water doesn't really fit through there very well. So the pores are small enough that it's going to catch all the bacteria. Depends, how small you have them as to what you're trying to catch. And then you hook this up to a vacuum to suck the water through that membrane with the really small pores. And that membrane is really thin. You're going to put that on top of some agar media. So the nutrients in the media will kind of diffuse up into that membrane. You incubate it, and then you count the number of colonies that grow up. And a lot of times there's a little grid printed on there to help you count that accurately. Okay, so and this way can be used. You can use certain media where certain organisms that you don't want to see in your tap water are going to turn a certain color. So there's a problem. There's some kind of bacterial contamination maybe with your drinking water. Okay, so both of those methods, again, they require incubation. So what's nice about that is that they are only going to count living cells. Those are usually the cells that you're concerned about. And with other methods, sometimes it's hard to tell if the cells are dead or alive. So that's one nice thing about this, both of these methods. And I would also add that both these methods are inexpensive. You don't need any fancy equipment in order to get your counts. Just, some media or Petri dishes or whatever, nothing too fancy. I'd say one drawback is that you're going to have at least an overnight incubation. So it's going to it's going to take a little bit longer. You're not going to get your counts back right away. Maybe that doesn't matter, but okay. All right, so moving on to some other methods that don't require incubation. So again, you can kind of get your counts back a little bit more quickly. If you had a sample that you are trying to test, you could take out an aliquot. Usually use a micropipetter to get a really small amount, and use something called a Petrov Hauser chamber. Okay, that's printed down there. We also call this a hemocytometer. Okay, I think on that slide there, if you look kind of really close, you can see that it's printed Petrov Hauser chamber. But basically, this is going to have a cover slip sitting like right here like this, and the sample that you're trying to count, you add with a micropipetter like into one of those grooves right there, and it will quick underneath the cover slip, but kind of a uniform depth. And then printed like right here and right here is a grid that you can see when you look at your cells under the microscope. Okay, and then you're going to zoom in on a certain number of squares and count the number of cells that you see. It has to be, again, within a certain range to be statistically significant. But knowing, how big of an aliquot you added there, you're going to extrapolate back to figure out how many cells were in your original sample. So that way is pretty quick. Again, you can't necessarily tell if your cells are dead or alive. Sometimes you can. It depends on what kind of cells you're using. Another method is using a spectrophotometer. So remember, a spectrophotometer, I didn't add any more. Basically is a way to see how much light can be either transmitted or absorbed through some sample. So it's basically a way to quantify the turbidity of our sample. So here's this tube you can see is a little bit cloudy. Okay, the absorbance is going to be higher for that tube than this other tube, which looks like it's just the media. You would have to kind of zero it out. Okay, here's what it is with just the media, because that has a little color to it. And then, okay, what's my absorbance on top of that? How kind of quantitating the cloudiness there. Okay, so, you know, obviously the absorbance is going to increase the more cells you have there. We don't get actual numbers from this though. You get an absorbance reading. So that's maybe one drawback. And also dead cells are going to scatter light just the same as live cells. But it is a nice way to see if you have a culture that you're trying to grow is getting, you know, cloudier and cloudier over time. So you can see if your culture is actively growing. Kind of quantitate that a little bit. Getting a little bit more on the expensive side now. Kind of getting a little bit, again, more and more equipment heavy. Something called a Coulter counter. This is an electronic cell counter. We do have one up in the labs that's, I think, the upper level class uses. But this works on the principle that bacteria are going to disrupt an electric field. So basically, if here's your sample there, you set the flow volume to be kind of a certain amount, and then as it passes through the electrodes, again, if there's a cell there, it will kind of make a mark of that. So it's really quick. Cell clumping can be a problem that can cause it to be not so accurate. And this also can't discern if the cells are dead or alive. But it is really fast, and you don't even need to do any calculations or anything. It just kind of tells you the answer. And then kind of the best of the best, the most expensive, if you really need to have your most accurate numbers, you use something called a flow cytometer. And if you happen to go to Dr. Renkema's talk on Friday, she uses this a lot in terms of trying to count different kinds of cell populations that she finds in her mice after she's infected them with something. But the way flow cytometers work, and there's, I should say, kind of little desktop models, and there's also like $1 million models. The one that she has is kind of somewhere in between there. But the way these work, you stain your cells with some fluorescent dye, and then the flow cytometer basically will count them based on, you know, whether they're exciting with that dye or not. And it also can separate out the cells that have a different kind of tag to them. So it's a really powerful instrument. The data that you get back is kind of difficult to decipher. But in this example, this data that I just show you as an example, it was some seawater that had been tagged with like 3 different fluorescent dyes. So there's 3 different kinds of cells that it recognized. And I forget, you know, the red, it was one kind of cell. Oh yeah, a certain kind of algae or plankton. The green was one and the blue was a different kind. So again, extremely accurate. Again, extremely expensive. You're not gonna have one of these in the dorm room, most likely. All right, so questions, don't be afraid to stop me. All right, moving on, thinking about different environmental influences that can affect growth rates and whether bacteria grow or not. And this is basically what next week's lab, we got a little bit ahead of the lab here, we'll talk about. We're going to talk about different physical influences. The first one that we always think about is temperature, because probably it's the easiest thing for us to kind of monkey with a little bit. But cells will typically have some ideal temperature that they're going to grow the fastest at. And again, that's variable. We have different terminology. We put labels on organisms based, and we'll talk about those labels in a minute, based on their ideal temperature. But usually, say this is the cell's ideal temperature right here. Usually, right after that ideal growth temperature, growth tends to bottom out. So, in other words, it just a little bit hotter over here. It's hot enough that enzymes are being denatured, and it just isn't going to support growth. So, okay. And a lot of times cells will be able to survive at different temperatures, but they're always going to, grow fastest at a certain temperature. So this is an illustration of, I forget, I think it was E. coli that was streaked out in 3 different plates. One of the plates was left at room temperature. One was a little bit warmer than that at 30, and 37 degrees Celsius is body temperature. So you can see the difference in growth. Excuse me. The one at room temperature only grew a little bit, and then faster and faster at body temperature, it grew quite rapidly, and you see a lot of growth there. So again, oftentimes cells have a range, but they'll grow quickest at one temperature. And of course, we like to label them. These particular terms are fairly intuitive. For instance, a thermophile is an organism that's heat loving, right? I wouldn't worry too much about the actual temperature ranges here, just that thermophiles typically will grow if body temperature is, remember, about 37 degrees Celsius. Thermophiles might grow best above body temperature to about 80 degrees Celsius. A hyperthermophile is one of those organisms maybe that would need a hot spring to grow in, like a Yellowstone somewhere that's hot all the time. There are even organisms that can grow near boiling, which is astonishing. Most of the organisms that we work with in the lab are considered mesophiles. Okay, they're going to grow somewhere between, if 20 is about room temperature, somewhere between room temperature and body temperature is going to be their ideal temperature. And then our psychrophiles, these are our cold-loving organisms that typically grow about room temperature and below. And then you can say, oh, our extreme psychrophiles are going to grow like on the polar ice caps and places like that. So basically, whatever the temperature, there's some bacteria that's going to be able to grow and thrive there. Same with pH. We put some labels on the pH that organisms are going to grow best at. And again, fairly intuitive. An acidophile grows best at low pH, a neutrophile at neutral pH, and an alkylophile, or sometimes alkalinophile at higher pH. So again, most of the organisms that we would work with, say in the lab, are going to be neutrophiles. Typically, organisms are going to grow over a rather narrow pH range. Again, they're going to just hydrogen bonds get and proteins denatured if you get into a different range. Examples of alkalinophiles or acidophiles. Acidophiles, like mine runoffs. Sometimes there are streams, for example, there are bacteria that actually can produce sulfuric acid, live at really low pHs, or there are bacteria in our stomach that can even grow at that low pH of like 2 in our stomach. And an alkalinophile that there's a bacteria, Vibrio cholerae, the bacteria that causes cholera, actually grows best at a pH of about 9. So some pathogens in there as well. Okay, so let's talk about oxygen. So just like organisms can grow at different temperatures or different pHs, different oxygen concentrations is another big one that we think about. kind of fine tuning if we're trying to grow that organism in the lab, or maybe we're trying to kill them. But there are a lot of organisms that don't need oxygen or are actually poisoned when they are around oxygen. So we'll get to their labels of what we would call them in a minute. But I want you to understand first that it's not atmospheric oxygen that's bad for organisms. It's not O2. It's the reactive oxygen species that is present in the atmosphere is what is bad for organisms. So we call them reactive oxygen species, or ROS. And here's some examples of reactive oxygen species. So I want to think about where these come from. Where do these ROSs come from? What's so bad about them? Why they do damage to cells, and what cells have to do to detoxify them. Okay, so those are three things that I want to talk about. First of all, what's so bad about them? I don't need you to understand the chemistry or write out the formula for these. In some cases, I don't have that memorized either. These are all strong oxidizing agents. And that means they are going to pull electrons from other molecules in the cell. So they are going to grab electrons from other molecules in order to fill up their outer shell. Okay, and that does a lot of damage. Okay, so they're strong oxidizing agents. Where do they come from? Well, a couple of these the cell is not likely to come in contact with, but singlet oxygen, that one is a result from photosynthesis when oxygen is boosted to a higher energy level. So plants have things like certain pigments to kind of counteract that. The two reactive oxygen species that we're going to be most concerned with here are superoxide and peroxides, because those are formed just as a normal part of aerobic respiration. And we'll talk when we talk about electron transport chain and aerobic respiration, we're going to talk more about where that actually happens. But sometimes oxygen actually has to split when it's the final electron acceptor to make water, and sometimes that doesn't split correctly. So you get superoxide, again, as a part of this about 2% of the time aerobic respiration instead of water. Hydroxyl radical cells don't encounter that too often because that happens during ionizing radiation. And hypochlorite ion, that is actually made in our neutrophils. Okay, in our white blood cells, we make hypochlorite ion, which is actually another name for bleach. Okay, we actually make a small amount of bleach in our cells in order to kill invading microbes. Okay, but let's zero in here on the superoxide and the peroxide. What do cells do to counteract that? Well, they make enzymes to detoxify them. So I don't memorize the reactions here, but superoxide plus 2 protons. And if a cell can make an enzyme called superoxide dismutase, then it can detoxify that superoxide there into hydrogen peroxide and oxygen. Okay, so now the problem there, hydrogen peroxide, okay, that's a reactive oxygen species as well. So cells have to have another enzyme to detoxify that. And one of them that's very common in cells is called catalase, commonly produced in cells, and detoxifies the hydrogen peroxide to water and oxygen. Just balance that. Okay, and that is a test that you're going to do if you are in lab later on in the semester. a catalase test to see if cells can detoxify peroxides. Most cells that live in aerobic environment are going to be able to make them. So, but some not. I know in my lab this morning, I talked about Bacillus and Clostridium. Bacillus is a genus of spore-forming organisms that are mostly aerobic. Clostridium, again, makes spores and anaerobe. How do you tell them apart? One way is to do a catalase test. And you take a glass slide, you rub your bacteria on there dry, and then you add a drop of hydrogen peroxide to it. And if it bubbles, like we see here, that's the oxygen coming off of that, then the organism must be making that enzyme to detoxify hydrogen peroxide. Okay, so Bacillus makes catalase and Clostridium doesn't. So it's just one way to tell some different organisms apart. There actually is another enzyme that some cells make to detoxify hydrogen peroxide, and that's called peroxidase. Actually, that involves an electron carrier. I didn't leave myself much space there to draw that in. Plus a proton plus NADH. Sorry. And then what we end up with is NAD plus H2O. Okay, I don't know if there is a test for peroxide. It'd be hard to test for these products, NAD and H2O. I don't know if there's a way to do that, but there are some organisms that can make peroxide. Again, it just detoxifies hydrogen peroxide in just a little different way than catalase. Okay, so the point is that we can categorize or put organisms in groups based on what it boils down to is whether they can make those enzymes or not. And this test you're going to do next week if you're in lab. It's called the thioglycolate test. That's written down here, thioglycolate tubes. It's a media where we have chemically gotten rid of the oxygen in that media because thioglycolate actually reduces the oxygen in there down to water. Okay, so it gets rid of the oxygen. Any broth, you would have less oxygen in the bottom than you would at the top, but this essentially, again, chemically gets rid of it. We also add an oxygen indicator in there, which they don't show here, where, the top part, where there is some oxygen, we'd always have a cap on here, but some oxygen diffuses under the cap, and at least the top part of the tube is going to have close to atmospheric oxygen. Okay, but then we inoculate our tubes, and we see where the organisms grow. If they only grow at the top of the tube where there's some oxygen, then we call that organism an obligate aerobe. If it only grows in the part of the tube where there's no oxygen, then we call it an obligate anaerobe. And then a facultative organism is one that grows pretty well, grows the fastest where there is oxygen, but can grow elsewhere as well. So facultative, that's another term for meaning flexible. Okay, organisms like E. coli that have flexible metabolism, they can respire aerobically. They can also maybe do some kind of anaerobic respiration or fermentation. We'll talk about those things starting next class. And then if you kind of split hairs a little bit, and this is kind of hard to tell from this test because it's not a perfect test and you can't always tell exactly where the growth is, but you might be able to tell if you had a microaerophile that's kind of growing, just kind of at the bottom edge that's oxygenated. So a microaerophile needs a little bit less than atmospheric oxygen concentration to grow. And then an aerotolerant anaerobe, this is going to be an organism that does not need oxygen for its metabolism. But it can detoxify those reactive oxygen species. Okay, so in other words, an example of an aero-tolerant anaerobe is a lot of our streptococcus. Maybe it's a genera you've heard of, maybe not. You've had strep throat, you've had streptococcus. But that's an organism, all the streptococcus do is ferment. And they don't care if there's oxygen around or not. They do this certain kind of metabolism that they don't need oxygen. They have those enzymes to detoxify any reactive oxygen species they may come in contact with, so they can be in the presence of oxygen, but they're going to grow the same anywhere in the tube because, again, they're not using the oxygen. Organisms that respire aerobically always are going to grow faster because they get more ATP when they respire aerobically. Okay, so so that's just a little introduction to oxygen requirements. And again, we kind of split hairs a little bit there. If you have an organism that maybe can't grow or doesn't have those enzymes to detoxify superoxide and whatnot, you have to grow them somewhere where it's not in contact with the atmosphere, basically. So a couple different ways we could do that. If you have an anaerobic organism, the organisms that we grew in lab today, the Clostridium, are they're anaerobes. They're a little bit tolerant of oxygen, so we had those out in the air. But when we grew them up in something similar to this. It wasn't quite that fancy, but it was a jar that we put a chemical pack in there to kind of have a reaction that got rid of the oxygen in there. When we opened that up, if you smelled something, if you were upstairs over there in Henry this morning, it didn't smell very good. It's particularly stinky organism that we were working with. So we grew them up with something like that. If you're really serious about working with anaerobic organisms, you might work in what we call a glove box, where those black holes, there's like a glove, you stick your hands in there and then you can inoculate your organisms that way. Okay. Kind of on the same lines, thinking about oxygen requirement, there are some organisms that are considered capnophiles. And that means that they grow best in an elevated carbon dioxide concentration. Okay, so way we often draw that is elevated CO2. When you put brackets around something, that means concentration of. Okay, so either they need more than atmospheric concentration of CO2 in order to grow, or maybe they just grow better when we give that to them. And that might be because they have a metabolic need for CO2, or just because they can outcompete other organisms a little bit better when it's around. So we have some organisms that we use in lab that are capnophiles. We have an incubator that's set up with a CO2 tank that we can provide that. A little lower tech way to provide high CO2 is with something called a candle jar. And if you're in lab, you're going to use this later on in the semester. Basically, you take a big old jar, maybe one that used to be a pickle jar or something like that, and you put your plates in it, and then you take a votive candle and you light it, put the lid on really tight. The candle releases some CO2 when it burns. And of course, it will go out as soon as it uses up all the oxygen in there. So yeah, we're getting rid of oxygen, but we're also putting a little extra CO2, and that gives our cells they grow a little bit better. Some certain kinds of cells that are considered capnophiles. Okay, so going beyond oxygen, just really a couple more environmental things that I want to talk about. Physical effects of water. So we talked about osmosis previously and reviewed that a little bit. This is one of the things that you're going to test in lab again next week. So osmotic pressure. So a lot of people, they don't really define this very well in your lab manual, so I wanted to define it here. A lot of people think about osmotic pressure as just being very different solute concentration than what's inside the cell. But if you really look at defining this correctly, let's call it the tendency of a solution to take in pure solvent by osmosis. So that means the solution is gaining water. Okay, so that's going to be a hypertonic solution. A water follows stuff. Okay, so if the water is going into the solution, there must be lots of stuff or solute in the solution. So when we talk about high osmotic pressure, we're talking about a solution that's very hypertonic. Okay, lots of solutes in it relative to the cell. And this is not what we mostly see in nature. Okay, mostly we see hypotonic solutions in nature, but there are certain places that we do see it, like the Great Salt Lake. And we are just out of time now, so we'll finish up with those definitions. I think I have one more slide with chapter 7, and then we'll start metabolism on Wednesday. All right, have a good day.